Today we're going to talk a little bit about the impact of having a high quality reference collection for doing the genomic and genetic studies. Excuse me. Oh, okay. So I'm not going really to talk a lot about the numbers and results, but the way we produce these reference collections and the methodologies that are available already in the market, the technologies that we use for producing these resources that can be referenced for conducting our studies. Few things that I really want to highlight is that, and I want to give this message to everybody that is dealing with these collections is, first of all, we need to define, we need to really pay attention to the quality of the collection that we are using. I've heard a lot from when we were talking with different researchers, when we talk about the genes or genomes, they say, oh, I downloaded this gene from NCBI as a reference platform, or I downloaded that genome from this dedicated genome reference collection from this group. But what we really need to pay attention is that even the reference collections, they are storing and collecting the data that was published by different research groups. And these data, based on the way that they conducted this analysis, the quality can vary. If they did a very good job, you will have a good quality reference collection in sense of, like, for example, genome assemblies and prediction of the genes. Or if they did a poor job, you will probably end up with having a genome assembly that the quality is not really well. I will also highlight a couple of challenges that we have in producing such collections, especially in the sense of the genome assembly and the prediction of the genes, the gene catalog in that. And also eventually I will introduce two examples in Carrot that somehow shows the impact of misprediction of the genes if you're using a low quality catalog for doing your investigation, and how it can impact your bias, actually, your analysis. At the beginning, what is the concept in the sense of the genome assembly? Simplified, we have the plant genome. We want to have that one in our computers in a way that the sequences, we know all of them, and we can manipulate that one. We want to characterize them, the genes that are on the body of the chromosome, and on downstream of the, and on the, at the end, we want to do some sort of like characterization and annotation of those genes, all the different components that we have on the genome within the cell. So later we can start to work on that one and use them as a reference for doing our analysis. Here, as an example, you see 9 carat chromosomes under the microscope, and as I mentioned, what we want these chromosomes in the sense of like sequence spaces in our computer genome browser characterized and annotated. So some of you might be familiar with the genome assembly process, but today, for those of you that are not familiar, I will provide a simplified summary of the workflow. It's not all of that. There are so many different steps involved, but in general, when we have the plant material, we want to do the sequencing. There are different protocols for doing the sample preparation and library preparation. I'm not going through that one, but the only thing that I want to highlight here is that for genome sequencing, there is no methodology or technique or any instrument available that you extract the genome, you send it to the sequencing facility or to that, using that instrument, that you can sequence the whole chromosome in one piece. There is no way you can do that so far up to now. So what is normally done is that after extracting the DNA, genomic DNA, they shear it, they fragment it, and prepare the library, send it to the sequencing facility, and using the sequencer, they start to sequence those DNA fragments. Soon after, we receive some files that are full of reads. We call them reads because they are the reads from these DNA fragments. So we have the bases sequenced, and they are coming randomly from all different parts of the chromosomes. These sequence files, this is actually the part that my job starts, and we proceed with the assembly of the contigs. There are two concepts I want you to pay attention to that, because I will use them a couple of times during this talk. One of them is the contig. the term contig. Contig means that when you put these sequences together and you don't have any gap in them, so you assemble them, you have a piece of sequence without gap, they are called contigs, so no gaps. And then by some means that I will introduce them later, we put them together because we want to reach to the chromosome scale sequence, right? When we put them together, and there are actually some gaps in between because there are regions that we are not aware of that, we call them scaffolds. These scaffolds later we use other means like genetic maps or other sequencing technologies, and we try to anchor them into the chromosomes. Genetic maps, I'm not going to explain how they are developed, or in general, it requires a complete session if you want to talk about them, but simplified, there are different markers across each chromosome, like labels, and they point to some positions in the chromosome. And we use those, we map them on these contigs and scaffolds, and by the order of the positions, if you sort them, you will have some sort of like contigs tiling the chromosome structure, okay? So that's what we use for scaffolding and putting the contigs and the scaffolds together and generating the chromosome scale assembly. And we call them super scaffolds or pseudochromosomes. I'm going to talk about these 5 steps a little bit more in details and the challenges that we face. Let's start with the sequencing. Actually, the sequencing phase started like on 97 with like, I think, with the Sanger sequencing. And the cost of sequences, as you can see here in this plot, was quite expensive. Like for a megabase of DNA sequence, you were going to spend like around $10,000, while during the years there was a tremendous effort on decreasing the cost, increasing the quality and resolution of the sequences, and also having a longer read comparing to the short reads. Right now, in 2019, you can simply sequence the megabases of the DNA or RNA with like few cents. And considering the human genome, you can simply sequence the human genome with less than $1,000. Not like with a very high coverage, but it is possible to do that. Talking about different sequencing technologies, you see here the different years and the time that these methods and methodologies were introduced to the market, and the throughput of them, because some of them have a faster, they have a higher throughput, some of them have lower throughput, some of them are like single sequence going through the sequencer, some of them have like hundreds and thousands of them going. So the NGS technology that was really a breakthrough at that time that was introduced, and still people are using that, the length of the sequence varies from like couple of 10 bases to 150, a little bit more or less. The Sanger sequencing is around like a couple of 100 bases to 1 kb. And then there were 2 technologies that were offered, one Oxford Nanopore and PacBio. These 2 sequencing technologies allowed us to go with way longer reads. And as you can see here, the read length is around 5 to 10 kb and 10 to 15 kb for these 2. There were also some other technologies offered like Dovetail. This methodology is not actually for producing the base, the nucleotide sequences to be used for the assembly, but it is mainly used actually for scaffolding purposes. So what it does in, actually, when you have the chromatin structure of the chromosomes intact, they go and start to label them. And normally the sequences that are adjacent to each other, they are likely coming from the same chromosome. After that, they open up the chromatin structure, they shear the DNA, and they start to do the sequencing of the ends of the read. Since the structure is unfolded, you will see that there is a big insert size in between, but these 2 actually are coming from the same chromosome. So if I have a dot here, I can say that this region is connected to this region, and for the dovetail data, for these parent sequence data, for 2 of the projects that we did before, the maximum insert size that we observed was around 7 megabases. So if the whole chromosome is like 50 megabases, one chromosome homolog. If you have these reads across that, you can simply put the contigs together and assemble that into the chromosome scale assembly. Going back to this one, there is also a bionano map that people are using for scaffolding process. This one also gives you a power of 100 to 250 kb distances that you can put the contigs and tile them together and generate your scaffolds. But what we lose and what we gain, when we are talking about short reads, since they are short, the quality of the bases are really high. So the NGS technology is like around 0.3% error rate. But when it comes to long reads, like here, the PacBio raw reads, as you can see, there are so many errors across these. And we have around 12.9% of error rate for PacBio. But the good thing is that these errors are random across the read. So if they're random, we collapse them together, we map them versus each other, and we start to correct them, those that are randomly happening. You will end up having the PacBio corrected reads, and what is presented for the PacBio technology is that the accuracy of the bases goes up to 99.9%. And there are some of them that you can see here. These are the polymorphism points, so they are not random, and we keep them later for our studies. Here also there's another demonstration of the Illumina short reads. As you can see, they are way shorter than the PacBio reads. And this one is an illustration of mapping them on the genome. But before having the genome, I'm just showing this one to show you the concept of the way that they are overlapping each other, because this is the main principle of doing the genome assembly. So going back to the sequence files that we received from the company, the sequencing company. To be honest, this is the best representation of doing a genome assembly. I couldn't find any better picture. You have a huge puzzle with millions of pieces, sequences, and you need to put it together. So the sequence files, based on the length of the sequence, look like this one. We have ATGC. We don't know where they're coming from. They're from different fragments of the different parts of the genome. And what is normally done is that, let's consider this example. We have these sequences. We start to compare them with each other. In general, the basic rule is to find for the similarity of the sequences and the overlapping of them. And then we try to put them together and create this consensus. You remember the contig concept? This is a contig. There is no gap in that. But, for the short reads, normally we have the read. There is a methodology that is used. This method is actually used a lot for the short read assembly as the De Bruijn graph. So they fragment the sequences into pieces. They call them k-mers, similar sizes. And then by overlapping them, create these graph paths and then put them together. There are tools like Trinity, Soap de Novo, Velvet that they are using this methodology. This is commonly used for short sequences de novo assembly. But for the long reads, the good thing is that they are way longer. The thing that we do normally is that we map the sequences versus each other and find the longest one, longest seed read. And this one is the pre-assembled read that represent a lot of reads. We first correct the errors, and then we proceed to do the overlapping and generate the contig. First barrier. We have millions of sequence fragments. And maybe it's not really relevant to the part of the scientific part that I'm going to present, but if you don't have a good cluster, it is almost impossible to do any of these things, the genome assembly, gene prediction, and so on. Just as an example, for Blueberry Genome, we had to run the assembly on main campus on 17 nodes of a cluster, and it took 2 weeks to finish. And we had to repeat that one for like 16 times to get the best assembly out of that. Some people are using cloud computing for that, but if you are really, like us, a lab that is really trying to find the best quality, run the assembly for couple of times and tune the parameters, that can be really costly, and sometimes managing the softwares on that is really difficult. So going back to the story of the assembly, the sequence similarity and putting them together, well, we can say, okay, that's simple. Anyway, it will take time, but we will find the similarity and overlapping of the sequences, put them together, and generate this consensus. So we will have the chromosome in this shape. But unfortunately, what we will get normally is something like that. We will end up having a lot of contigs. When this is not something that the assembler output, but if you want to proceed with the assembly graph, the similarities that you find in the assembly and putting them together, this is the representation of that one. So why this is happening? Why we cannot have the whole sequence in one piece? One issue. The challenge number one that I'm going to present is low coverage. It's possible that the DNA is degraded. Some of the DNA fragments are degraded. You don't have coverage on some regions of the genome. It's possible that it didn't go through the sequencing or whatsoever. Based on any reason, you don't have coverage in that region, and when you're putting them together, when the assembler reaches here, it drops. It thinks that it's the end of the chromosome. There is no other evidence to proceed. There is no overlapping. So this is one reason that we have the contigs, and they are not spanning the whole chromosome. The other issue is the repeat content of the plant genomes. Actually, plants are full of repeats. And if you look at this example, this is blueberry diploid genome. We have over 30% of the genome sequence with aggressive repeats, and up to 70%, more than 70% with like total of simple and aggressive repeats together. So this repetitive sequence is actually causing another challenge in putting the sequences together. Let's see what happens. In simple words, we have this genomic region. We have these two repeat copies. They are similar. So when the assembler comes here and put the reads together with this single copy, when it reaches here, there are different possibilities, because now we ordered them and we are just showing them, but when the assembler is dealing with them, it doesn't know the order. If we are lucky, it goes past A. So we have all these three assembled correctly together. If you're not lucky, it goes B or C. So you can have either miss this part, or you can have a misassembly and misjoin of the sequences together. Unless you have a very long read that is spanning this repeat region and is coming from the single copy to single copy, so the assembler can pass it, which sometimes the repetitive regions are quite large, and still we don't have that long sequences that can overcome that. Here, we call it the repeat fork, and in other words, when you have the single copies, they come to the repetitive sequence and the assembler gets confused. Okay, which one to choose to go? And that's why, well, assemblers normally drop it there. They don't proceed any further unless there are enough evidence or it gets confused and create the misassembly. So what we get as an output of the assembler is a whole bunch of contigs that are not spanning the whole chromosome, but they are way longer than the reads that we use for the assembly. Okay? There are techniques already available that we can use for putting them together. One of them is using the parent data and the technology that I was mentioning before, the PHI. So we have these two parent sequences with the long insert sites. So if we have a lot of contigs and there are reads that are falling in the single copy regions of the contigs. Look at this, for example, we have reads that are spanning from this contig to this contig, and this one to this one and this one. So we can put them together, but still, obviously, we will have still some contigs that cannot be scaffolded. That's why in the genome assembly files you have scaffolds, super scaffolds, and still you have some contigs that are left over and they could not be anchored into chromosomes. The last challenge I'm going to mention in the face of the assembly part is for diploid and polyploid genomes. So as a quick introduction to the polyploidy, if you have a haploid genome, you have one copy of each chromosome for that. But if you have a diploid genome, there are two copies for chromosome 1, for example. So these two copies are homologous chromosomes. And if it's a highly homozygous genome or is a double haploid, they should be identical or very similar. But if it's a heterozygous genome, there are regions along the body of that chromosome that are different, and we call them heterozygous sites. Imagine when you go to the tetraploid, triploid, tetraploid, hexaploid, octaploid, and the poly level increase. So what problem this one caused in our genome assembly? If you look at that one in this sense, you can see that there are homozygous sites that are similar. There are sites that are heterozygous, so they are different. And the assembler, when it's doing the assembly, so what makes them heterozygous is that there are structural variants, SNPs, polymorphism in those regions. When assembler is going through them and it wants to put them together, what happens is that these homozygous sites they are similar. It puts them together and consider as the similar reads coming from the same region. But when it comes to heterozygous sites, it assembles two different sequences, right? So in reality, what we expect actually, since we know it's a diploid genome, we are expecting that the output of the assembly is two sequences for each haplotype, and the heterozygous sites are correctly phased. So those that are coming from one chromosome are in one sequence, and those that are coming from another one are in another one. But what happens normally is that we have a lot of misspacing, because again, like repeats is this different concept, but the procedure is similar. It comes to this region, there is similar, it doesn't know which one to choose, can go through. So puts one of them in the sequence structure. So this one also caused another issue. And there were methods published, and people are using that. One of them, in brief, is Kannapolis. You need to have the sequence data from the parents for the individual. And what they do, they create sequence-specific clusters for each parent. So the sequences that are specific for each of the parents. And then they use those for separating the haplotypes in the offspring. Another method is, again, using the Hi-C data. I told you those are long-distance parent sequencing data. And if they are falling on the heterozygous sites, we can use them for doing the phasing and putting the correct phases together and having the homologous chromosomes correctly phased. So now that you know that you got this information about the barriers that we really face, three of the major barriers we face in the genome assembly, I would like to give you a little bit of information about two of the crop species that we did the genome assembly on them. We are highly interested in these two. Our lab is working a lot on these two crop species. And as for the blueberry, I will talk a little bit about the assembly process. But on the carrot, I will proceed and talk a little bit about the gene prediction and the quality of that one. Since I did a lot of, well, in our lab, we did a lot of work, manual work also in validating the genes and the structure and this kind of stuff. So there are some examples that really transfer the meaning of having a high quality reference collection. So as for the blueberry, we sequenced the blueberry Vaccinia caesariensis. It was, the genome size was around estimated to be 673 megabases. It's a diploid genome, highly heterozygous, so we have regions that are completely different in the homologous chromosomes. And the target was to have a high quality haplotype resolved chromosome scale assembly. There was before one genome assembly of the blueberry. It was highly fragmented, and the bases that are sequenced is, I think the proportion is around 60% of the total genome size. The gene prediction was also quite poor. And what we wanted to do is to improve the quality and having a chromosome scale assembly, haplotype resolved, so two homologs sequenced independently and assembled. As for the carrot, Dacus carota, the genome of this carrot was published in 2016 by Massimo. And this genome is highly homozygous. It's a double haploid, so the homologous sequences are almost identical. And although it's a diploid genome, the homologous sequences can be phased as like a haploid sequence because they are similar, so we can proceed and we don't need to be worried about the structural variant and polymorphism across that one. So what was the target was to improve the base pair quality of that one, since the genome assembly that was done on 2016 was using the Illumina sequencing, short reads. So it was highly fragmented. Although it was anchored into chromosome level, but we have a lot of gaps across the sequences. And also we wanted to improve the gene model prediction using the long read sequencing. As for the blueberry, we sequenced 54 cells, 54 runs of the sequencing, and it was the RS2 chemistry. If you want to know a little more about that one, you can go to the PHI website and read about this one. But it's an older version of the smart cell, and we generated around 2 million raw reads with the 53 gigabases of base pair sequences, which was around 80 pair coverage of the whole genome, and the total and the average read length of 10 kb. As I mentioned earlier, we ran around 16 rounds of assembly in different cases, tuning the parameters. We started to get like different genome sizes, and eventually in the final round, after optimizing that one, we got something close to the estimated genome size. And using the high-C data that I mentioned earlier and Falcon phase pipeline that was offered by PHI company, we were able to phase this genome into 2 haplotypes for each chromosome, phase 0 and phase 1, and the total length of phase 0 is 702 megabases, and the other one is 643, with the N50 value of 494 kb, around 490 kb of the phase 0 and 420 kb for the phase 1. Using the high-C data, not only we were able to scaffold it, we were also able to break a lot of misassemblies, over 500 points of misassembly in these 2 phases, because the connections were in a way that were confirming there is no connection there, so we were able to break that one and correct the misassembly points. And now we have these 2 phases in 12 chromosomes. We did a verification also using the genetic maps. As you can see, we have the diagonal in all of the cases, so it's the genetic map coordinates versus the physical coordinates of the scaffolds. For both phases, we have them anchored, but there were still some cases of the misassemblies that we should address them and proceed with the gene prediction. So we were able to produce the haplotype resolved chromosome scale assembly for blueberry, and that was the target, and we are proceeding for the downstream analysis on that one. Moving to carrot, since I really want to highlight a couple of interesting things in that one. We did the same sequencing, but different chemistry, 8 SMART cells. And we ended up, this was a newer version, so the yield was way higher. We got 4 million sequences, 34 gigabases, around 72 per coverage of the genome, and the average read length was 8.4. We proceeded with the assembly. So this is the one that was published in 2016. As you can see, each of these squares shows the proportional length of the contigs in the assembly. And when you compare them with the Canoe, this is the software that we used for the assembly, you see the proportions, how different they are. So this is the contig, this is in the contig level, so there is no gap in that one. So we ended up with the 436 megabases in the length, and the N50, that is a measure of assessing the quality of the contiguity, was improved over 100 folds. We also were able to add around 49.6 megabases of extra known sequences thanks to the long-read sequencing to this assembly. In another illustration, just to show you, there are these. This is the black one is the contigs that we have in PacBio, and there are 24. So the whole chromosome 1 is a scaffold assembled into 24 contigs, no gaps. And the gaps that you see here in between are between the contigs. This one is the V2 contigs. And as you can see, this is clearly showing the fragmentation of these contigs comparing to the PacBio assembly. You can see, as for the repeats, many of them were overcome. I'm not sure if this is really clear, but when we have the peak of the repeats here, those are the places that the contig is dropping, confirming the challenges that I was highlighting before. We also mapped the genetic maps. We had different genetic maps for carrot, mapping them on the contigs and scaffolds. Just as an example, for the linkage group 1 that represent one of the chromosomes, as you can see here, we have this piece, this piece, this piece, and a couple of others. If we put them together, these sequences, and sort them by the centimorgan, the genetic map location, we were easily able to anchor and also orient these sequences and build up the whole chromosome. Moving to the gene prediction. So now that we have the high quality continuous chromosome scale assembly of the carrot with less gaps, we proceeded with the gene prediction to create the gene catalog out of that one. And I'm not going to name all of them, just 2 major technologies for the sequencing of the RNA that is really used for gene prediction. Using the Illumina techniques, the short-read sequencing, as we already talked about that one. But the concept is the same. You extract the RNA. Well, the protocols are different, but the concept of shearing the RNA, sequencing that one with the Illumina technology, and then assembling the transcripts together to have the gene models. This is what we do with the Illumina sequencing of the RNA. But with the long-read sequencing, there was another type of sequencing for RNA, actually. The one that we talked for PHI was for the DNA sequencing. The PHI also offered a iso-seq sequencing. That this one, they do some sort of like, they put primers or like hair clips around the RNA without shearing that, and they go for the sequencing of long reads. So in that sense, they have multiple copies of the same RNA going around, and they produce the full-length transcript consensus for each mRNA that was sequenced. Happily, we're lucky to have access to, so here is a demonstration of the iso-seq mapping on the genome. You see how clear and the gene structure is very well characterized. Here is the Illumina gene models. In some cases, yes, they are confirming, but there are also some that are expanding. This is a misassembly of the transcript, and we have it that is expanding and connecting also these two genes together. We were lucky to have access to this data. Our lab produced that iso-seq transcript collection, and the Illumina data, we produced some, and also we had access to the publication of 2000. We downloaded some from the 2016 publication. We downloaded and used also some EST collection from NCBI to use as the experimental evidence. So these were the experimental evidences, those that were really coming from the RNAs to be used. And then we also used some other genomes that the prediction was conducted on them for the homology-based methods. Kiwi fruit, because we knew that the gene prediction in this one was all of the genes were manually curated, so it was a great set to be used for the homology-based technique. Ginseng, a close relative to carrot. Lectuca, the lettuce genome prediction, gene prediction. Arabidopsis thaliana, and Solanum lycopersicum. We predicted 33,777 gene models in the new assembly, which is around 1,664 more than the V2. So is that all? Did we really only predict more? What are the differences between the V2 versus V3? The one that we predicted now, we call them V3, and the previous one is V2. So let's have a look. I went through, like, I can say around 4,000, 5,000 gene models for validating and doing also a couple of verification of the gene models. And what I was facing and not really looking for them, it was coming up when you're browsing the genome browser. As an example here, this is the gene model that is predicted on the new genome, V3, and this is the old version, the previous version that we had, and these are the experimental data. As you see, the gene is partially predicted, and a lot of exons and that are not even predicted. So we have the experimental evidence, so we are sure this is a correct structure, and multiple records are confirming that one. In another example, this gene was predicted as one gene, but actually these are two genes, and they were linked together, and we have experimental data confirming that one in iso-seq and also in Illumina. Opposite side, we had genes that were split. So it was predicted as two genes, but it was one gene. And now with this long-range sequencing of the mRNA, we have confirmation that, yes, this is a true model and we can trust it. Another example of the split genes, actually there are 3 genes. And we have also cases with 4 genes, and I think 1 with 5 genes. In this case also, this exon is completely skipped in the middle and is not predicted. So if there is any expression analysis and so on, you're going to miss this part. Now that we talked about the gene prediction and the biases that you saw, the issues that you saw in the prediction of the gene models in the assembly, let's see what advantage this new gene prediction gave us in conducting our study in genetics, in carrot genetics. I'm going to end my talk with 2 examples. Our lab is interested and working a lot on characterizing the anthocyanin accumulation in carrot as well as some other crops. There has been 4 different regions that were associated to the anthocyanin accumulation in carrot. And this RAA region is mapped to coordinate and somehow balance the production of different types of acylation, the acylated and non-acylated form of the anthocyanin. If we look at the P3 region, so there were 33 genes annotated, and none of these genes were overlapping with the anthocyanin QTL. So we had no gene annotated that was none of these genes were actually overlapping with that region, indicating that they're playing a role in the anthocyanin accumulation. If we look at that region, a region of 494 kbs, there are 29 genes there. None of them are flavonoid structure genes. There are 7 MeBHP-like genes across that region and 7 BHLA transcription factors. So in the V2, in this region, there was no gene, and I'm sorry, I missed this part. And on 2017, there was this DCMIP6 that was reported by a Chinese group that is playing a role in anthocyanin accumulation. And as you can see here, the expression associated to the phenotype, but we were not able to identify that one in that region. The gene was not there. So in the new gene prediction and the new genome assembly, this is the V2, this region, the gene was not there, but now in the V3 version, the new gene prediction on the new genome assembly, the gene is there. And with the further investigation, we saw that this cluster of MeEPs that are here is a cluster of MeEPs, and they are in tandem, there are repeats, and probably that was the reason that the Illumina sequencing, the assembly of the Illumina sequences was not able to completely assemble that region, and the gene was missed and was located in an unanchored contig. The RAA locus that I introduced at the beginning, this one is mapped that regulate the different types of anthocyanin, acylated versus non-acylated. And in this fine map region, 370 kb, no anthocyanin-related gene was predicted in V2. In the V3 version, saw that the gene, there are genes that predicted in that region, that there is no prediction in the V2 for those. And this one is, so now we have candidate genes that we can further investigate and see if they are the genes that are controlling this RRA locus. By that, I want to conclude this talk and raise again this issue that when we are going to deal with, when we are going to conduct studies and analysis on that include the reference collections, reference genomes, reference gene predictions and annotations, the important things that we really need to know and always be cautious about that one, how the assembly, the gene prediction was done, what data was used, and always be cautious of the possible misassemblies, misanchoring, mispredictions, and also sometimes misannotation of these components in the genome that really can bias your analysis. You saw 2 examples. The genes were not there, they were missed, and we were missing them in that region. By that, I would like to thank you all for your attention. Also, thanks Plants for Human Health Institute really for providing us these facilities and resources to conduct our research. This is a picture of our lab, actually some PTP students, and on a daily basis I'm in interaction with them, talking about different topics. It's a learning process for me. And again, thank you so much for your attention.